Welcome back to the Talking Ketamine podcast. This is episode 46, The Science of Dissociation.
Glad to be here. Today we are getting into the weeds, or maybe I should say into the cortex. We're looking at a brand new 2025 review paper that asks a huge question. Is dissociation just a weird side effect? Or is it the point?
And that really is the central question, isn't it? Especially for anyone who's actually been through it.
Absolutely.
Ketamine was introduced way back in the 60s. It was supposed to be a safer version of PCP and anesthetic.
Right.
But it came with this very unique state of consciousness. Sensory detachment, feeling disconnected from your body, time and space just warping. And they called that dissociation.
And for decades, that was just seen as a bug, not a feature. It was something you had to put up with to get the anesthesia.
Just a nuisance?
Exactly. But now, with all the new tools we have, the whole perspective is shifting. We're seeing those effects as, well, not just clinically important, but maybe mechanistically vital.
So that's our mission for this look into the material, right? To figure out what the brain is actually doing when it enters that state.
Yes, and we've got a great guide. We're leaning on a big 2025 review paper by Bera and colleagues that pulls everything together: pharmacology, imaging, systems, neuroscience.
All to define how ketamine changes those cortical circuits. We're talking about NMDA receptors, HCN1 channels, really getting into the nuts and bolts.
Let's start right there with the basic chemistry. We know ketamine is an antagonist. It blocks the NMDA receptor, which is, key for learning and memory.
But how does just blocking a receptor make you feel like you're floating outside of your body? That's a huge leap.
It is a huge leap. And the answer is that it's not just a blanket blockade. It's much more subtle than that.
Okay.
This is where it gets really elegant. Ketamine preferentially blocks the NMDA receptors on very specific cells. We're talking about the fast-spiking inhibitory cells.
The GABAergic interneurons. Like PV and SST cells.
That's them. Think of them as the brakes in the system, the traffic cops. They keep everything orderly and controlled.
So ketamine isn't hitting the main gas pedal, it's hitting the brakes.
Precisely. For various reasons, the NMDA receptors on those brake cells are just more vulnerable to ketamine than the ones on the main excitatory neurons.
So if you block the brakes, the result is more go. It's disinhibition.
It's net disinhibition, exactly. You get this enhanced activity in the downstream pyramidal neurons. But here's the cool part. It's not just chaos. It's not just turning the volume up to 11.
It's something else.
It's a total reconfiguration. Researchers call it the switch.
The switch. Okay, tell me about that.
What they see is that the groups of neurons that are normally active when your brain is just idling along, they go silent.
To get turned off.
And at the exact same time, neurons that were previously dormant, just sitting there quietly, they suddenly fire up.
So the total amount of brain activity might be the same, but it's coming from completely different places.
You've got it. The volume is stable, but the station just changed completely. The whole network gets fragmented and starts forming these new, complex patterns that are pretty much independent of what's coming in from your senses.
The brain is basically having a conversation with itself.
It's rebooted its own internal operating system, and that internal conversation is the subjective experience of dissociation.
Okay, so if the network is fragmenting and turning inward, how does that create the specific feeling of, say, time distortion?
That brings us to the signature of this state, the traveling wave.
A traveling wave.
Yeah, researchers, particularly a group led by Vasuna, they found this very specific, very slow rhythm. It's a 1 to 3 hertz wave of activity.
1 to 3 hertz seems incredibly slow for brain activity.
It is, and that slow speed is the key. This wave starts in a really important hub area, the retrosplenial cortex, or RSC, layer 5 neurons specifically.
And the RSC is involved in memory and our sense of place.
Right. It's a critical hub. And when this slow wave takes over, something profound happens. The RSC stays connected to deep, old structures like the thalamus, but it becomes functionally uncoupled from most of the rest of the thinking cortex.
Wait a minute. So you're saying that feeling of detachment, of being disconnected from your senses, we can see it. It's that uncoupling.
It appears to be the direct neurophysiological basis for it. The uncoupling is the dissociation. We even see it in how sensory information is processed.
How so?
Ketamine doesn't block the initial signal. The early parts of a sensory potential, the P1, the N1, they're fine. The brain sees the stimulus.
It gets the raw data.
It gets the raw data, but it loses the later parts, the P2, N2, P3. And those are the feedback signals. They're what provide context and meaning.
So it's the difference between seeing a clock and understanding what time means to you in that moment.
Exactly. You get the stimulus, but you lose the story that goes with it. The result is an experience completely untethered from coherent reality.
That makes so much sense. Now, you mentioned a second target earlier. The NMDA receptor blockade starts the switch, but what about this HCN1 channel?
Right, so the NMDA part is necessary, but it's probably not sufficient. You need this second player, the hyperpolarization-activated cyclic nucleotide-gated channel, HCN1.
I always think of HCN1 channels being like the pacemaker for a neuron, sort of setting its rhythm.
That is a fantastic analogy. They're crucial for excitability and rhythm, and they're packed into those same layer 5 neurons in the RSC where the wave starts, and ketamine inhibits them.
So let me see if I have this. The NMDA blockade takes the brakes off, causing the initial disruption, and then the HCN1 inhibition comes in and helps sustain that new strange rhythm.
You've summarized it perfectly. It amplifies and stabilizes the new state. In fact, the research shows that HCN1 activity in the RSC is required for ketamine to create that slow wave and the dissociative behaviors.
So this isn't some messy side effect. It's a precise, multi-target neurological event.
It really is.
Which brings us right to the big clinical debate. The question every patient has. Is this whole intense experience, the trip, just a side effect? Or is it actually necessary for the medicine to work?
And for a while, the data pointed very strongly to it being a necessary biomarker. Meaning studies from people like Luckenbaugh and Fava, they found a direct correlation. They used dissociation rating scales, the CASA scores, and saw that the more intensely a patient dissociated,
The better their antidepressant response was.
the greater and more sustained the response, yes. The feeling was like proof of concept. It showed the drug got in there and hit the target hard enough to flip that switch.
Okay, so that's the biomarker argument. But you said for a while, what changed?
The masking studies changed things. They asked a brilliant question. What happens if you give someone ketamine while they're under general anesthesia so they can't consciously experience anything? The results were stunning. In both animal and human studies, when you co-administer ketamine with something like isoflurane, you block the conscious experience, you suppress that neuronal switch, and you completely block the rapid antidepressant effects.
Wow. So just having the chemical in your brain isn't enough.
It seems not. The brain needs to be awake and actively processing that reorganization for the rapid therapeutic benefits to actually take hold.
The conscious experience itself might be part of the therapy.
It strongly suggests it.
Okay, that's incredible.
Yeah.
But it raises another huge question. The dissociative part lasts, what, an hour? But the relief can last for weeks. How? The drug's half-life is super short.
This is where we go from the immediate network event to the long-term cellular repair. And it starts with a little trick called ion trapping.
Ion trapping.
So ketamine is a weak base. It can slip across the cell membrane very easily in its neutral form.
It gets inside the neuron, no problem.
Exactly. But once it's inside, it finds these little acidic pockets organelles like lysosomes and synaptic vesicles. In that acidic environment, ketamine picks up a proton, gets a positive charge.
And a charged molecule can't just slip back out through the membrane. It's trapped.
It's trapped. So even as the drug disappears from your bloodstream, you have this high concentration just sitting inside your neurons, slowly being released over a much longer period.
And that gives it time to work.
It gives it time to engage in deep, long-term changes through epigenetics.
So changing how genes are expressed without changing the DNA itself.
Right. It does two main things. It increases histone acetylation, which is like loosening the spools that DNA is wrapped around, making genes easier to read.
Opening them up for business.
And second, it changes DNA methylation. Specifically, it reduces the methylation on the promoter for the BDNF gene.
Brain-derived neurotrophic factor. That's the miracle grow for the brain you always hear about.
It's absolutely vital for building new connections. So by removing that methylation block, ketamine turns up the production of BDNF. And that leads directly to what we can see under a microscope weeks later. New dendritic spines, new synapses, new connections are getting built.
So the dissociation is the trigger. It's the short, sharp shock that starts this long, slow process of physical rebuilding.
That's the model. The brain disconnects itself in order to rebuild itself.
It's an incredible picture. Disconnecting to reconnect, literally.
And understanding this whole cascade, from the switch to the wave to the epigenetic repair, it points us toward the future.
How so?
Well, we have to remember ketamine isn't just one molecule in the body. It gets metabolized, it breaks down into other things like norketamine and, fascinatingly, hydroxynorketamine, or HNK.
And are those metabolites active too?
They are. And what's so compelling is that HNK seems to have very little NMDA blocking activity. It doesn't cause much dissociation.
But it still works as an antidepressant.
It still shows strong antidepressant effects, but through a different pathway, acting on the TRKB receptor, the same one BDNF uses.
So we might be able to get the long-term repair without the intense, acute dissociative experience.
That is the hope for next-generation treatments, to isolate the molecules responsible for the neuroplasticity from the ones causing the intense conscious shift. We're moving from a magic bullet to a whole cascade of targeted mechanisms.
Which leaves us with a really provocative idea, doesn't it? The link between a conscious alteration of the mind and the physical healing of the brain. Is that altered state the necessary price of admission for that kind of profound repair? It seems like we're finally getting the tools to answer that. Thank you for joining us for this analysis of the source material.